Welcome to Chapter 2, Sequence Analysis and Machine Learning. In this chapter, we'll learn practical protein function prediction using computational methods. You'll discover how to search for similar sequences, extract meaningful features, and build machine learning models to predict protein properties. By the end of this chapter, you'll be able to execute blast searches to discover homologous proteins, extract physicochemical features from amino acid sequences, predict protein localization and function using machine learning models, predict enzyme activity using random forest and light GBM, and evaluate and improve model performance. Let's start with sequence alignment. Sequence alignment is a technique for comparing two or more sequences to evaluate their similarity. Given a query sequence, an alignment algorithm compares it against a database to find homologous sequences. These homologs can help us infer function and analyze evolutionary relationships. BLAST, which stands for Basic Local Alignment Search Tool, is the most widely used sequence similarity search tool. With BioPython, we can programmatically run BLAST searches against NCBI servers. We specify the program type, like BLAST for proteins, the database such as the non-redundant database, our query sequence, and how many hits to return. Here's what BLAST results look like. For each hit, we get the sequence description, the e-value which tells us statistical significance, the score, and the identity percentage. The alignment shows matching residues with vertical bars. An e-value of 2.5 times 10 to the negative 75 indicates a highly significant match. There are two main types of alignment. Global alignment, using the Needleman-Wunsch algorithm, compares entire sequences end-to-end. This is best for sequences of similar length. Local alignment, using Smith-Waterman, finds the most similar subregions. This is what BLAST uses, making it effective for finding conserved domains even when overall sequences differ. Before we can apply machine learning, we need to extract features from sequences. The 20 standard amino acids have different physicochemical properties. We can group them by hydrophobicity, polarity, charge, and aromaticity. Understanding these properties helps us engineer meaningful features for prediction. Amino acid composition is a basic but powerful feature. We count each amino acid and convert to percentages. This gives us a 20-dimensional feature vector. We can also aggregate into higher-level properties like percent hydrophobic, percent charged, and percent polar. These aggregated features often correlate with protein function. K-mer representation captures local sequence patterns. We slide a window of size k across the sequence and count each unique substring. For k equals 3, we get tripeptide frequencies. This is analogous to n-grams in natural language processing. The feature vector dimension is 20 to the power k, so keep k small to avoid dimensionality explosion. The Kite-Doolittle hydrophobicity profile reveals protein structure. We calculate average hydrophobicity in a sliding window. A short window of about 9 residues highlights local surface character, while the 19 residue window from the original Kite and Doolittle paper is the one used to screen for transmembrane segments. Positive values indicate hydrophobic regions, negative values indicate hydrophilic regions, and with the 19 residue window, a sustained score above about 1.6 suggests a transmembrane segment. This profile helps predict membrane proteins. Let's apply these features to predict subcellular localization. Proteins function in specific cellular compartments: nucleus, cytoplasm, membrane, mitochondria, or secreted pathway. Each location has characteristic sequence features. We can train machine learning classifiers to predict localization from sequence features. We'll use random forest for localization prediction. Our sample dataset has 500 proteins across 5 locations. After training, we examine feature importance. Hydrophobic percentage is highly predictive because membrane proteins are rich in hydrophobic residues. The confusion matrix shows how well we classify each location. The classification report provides detailed metrics. Precision tells us what fraction of predictions for each class are correct. Recall tells us what fraction of actual instances we found. F1 score balances both. Membrane proteins often have the highest scores because their sequence signatures are most distinctive. Now let's tackle a regression problem, predicting enzyme activity. Enzyme catalytic efficiency is measured as kcat over km. We'll predict the log of this value from sequence features. This has practical applications in enzyme engineering and drug metabolism prediction. We extract a comprehensive feature set from each enzyme sequence. All 20 amino acid compositions, sequence length, physicochemical properties, and molecular weight. This gives us over 20 features per protein. More sophisticated approaches might include K-mer frequencies or position-specific features. For this regression task, we'll use LightGBM, a gradient boosting framework. It builds trees sequentially, with each tree correcting errors from previous ones. Key hyperparameters include number of estimators or trees, learning rate which controls step size, and maximum depth which limits tree complexity. Here are our prediction results. The scatter plot shows observed versus predicted activity values. Points close to the diagonal line indicate accurate predictions. On this illustrative dataset, we reach a mean absolute error of roughly 0.3 log units and an R-squared near 0.85. Treat those as demonstration numbers. Real enzyme kinetics data is far noisier, and published sequence-only models typically score well below this. To ensure our results are reliable, we use 5-fold cross-validation. We split the training data into 5 folds, train on 4 and validate on 1, rotating through all combinations. The mean CV score with confidence interval tells us expected performance on new data. This guards against overfitting to a single train test split. We can further improve performance with hyperparameter optimization. Optuna is a powerful framework for this. We define a search space for each hyperparameter, and Optuna intelligently explores combinations. After 50 trials, we usually land on settings that beat the hand-picked defaults, though how much they gain depends heavily on the dataset. Let's summarize what we've covered. First, sequence alignment using BLAST for homology search, with both local and global alignment options. Second, feature extraction including amino acid composition, K-mer representation, and hydrophobicity profiles. Third, machine learning using random forest for classification and LightGBM for regression, with Optuna for optimization. Key takeaways to remember. BLAST is your first tool for understanding unknown sequences through homology. Feature engineering from sequences is essential. Think about physicochemical properties. Machine learning models can predict protein function with high accuracy. Always use cross-validation to assess reliability. Don't skip hyperparameter tuning, it makes a real difference. Watch out for common pitfalls. Don't confuse BLAST E-values with P-values, use application-appropriate thresholds. With K-mers, keep K at 2 or 3 to avoid dimensionality explosion. Regularize models to prevent overfitting. Handle imbalanced datasets with class weights. Always normalize features by sequence length when comparing proteins. Here are the key tools we've used. BioPython provides BLAST interfaces and sequence utilities. Scikit-learn offers a wide range of machine learning algorithms. LightGBM excels at gradient boosting for regression and classification. Optuna handles hyperparameter optimization elegantly. All are open source with permissive licenses. In the next chapter, we'll dive into molecular docking and interaction analysis. You'll learn to use AutoDocVina for predicting how small molecules bind to proteins, visualize binding sites with PyMol, and apply graph neural networks for binding affinity prediction. Get ready to explore the 3D world of protein-ligand interactions.